All right. Yeah, so day 2 of the vision system. We have, so we left off nine less slides today, so we go a little bit slow. All right, so we left off talking about transduction, visual stimulus, transduction, and everything that's going on in the retina, but we haven't even left the retina yet, right? We're just at the level of the optic nerve. So what are the role, who can tell me what the role is of horizontal cells? Horizontal cells in the retina. What do they do? What's that, Lily? They make the edge sharper, and how do they do that? Yes? Lateral inhibition, right? So horizontal cells are responsible for lateral inhibition. Why do we need lateral inhibition? To make edges stand out more, right? Now, someone else tell me what amacrine cells do. Yes, Katie?
Are they the one that create the double signal with that possible, where you have two signals, one excitatory, one inhibitory?
Yes, they are responsible for creating two signals, an excitatory and inhibitory signal. Why are they doing that? What is the point of doing that? Yes, convergence, right? We have way more receptors than we have axons that can go to the brain, so we have to funnel hundreds of receptors into a single ganglion cell. And when we do that, we lose the ability to tell where light is in that receptor field unless we create two signals, and then we can at least tell if the light is in the center or the surround, right? So amacrines are responsible for center surround receptor fields that give us two signals for the price of one neuron. Very good. Okay. Mach bands. These are what's called Mach bands. These are, I would say they're an illusion. Again, going back to Plato, we don't want to trust our senses. So at point D and at point B, what do you see on this picture? What do you see at point B? That's probably easy. Like a bright line, right? And what about point D? Is there a darker line there? Okay. In reality, this is just going from dark gray to light gray. That light color is not really there, but you can cover it up and see that it's not there anymore. That's because of lateral inhibition. We're back to lateral inhibition. As this is going from darker, less inhibition, darker band, more inhibition, lighter band, right? Instead of it looking like this, dark, 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 increasing intensity to lighter, right? What we end up getting is this. We get dark, 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 and then you have that edge, that bump down, that dark band, and then transitions, and then when we get over to this edge, we get a slight bump up, and so you see that light band, right? Okay. Another example of how lateral inhibition is working. All right. Okay, we have two eyeballs, which means we have binocular vision. We have binocular vision, right? So there are two images being projected, but you guys are seeing one image of me, right? Everybody sees only one, not two of me. That would be a problem. We have to talk about that. Okay. So that is because the image coming into your left and your right eye get consolidated into a single place, into the same place in your visual cortex, so that those two things become one object in your brain, right? And so this happens, so if I have something over here in my right visual field, right? It is coming in and hitting the nasal portion of my right eye and the lateral portion of my left eye. And I have to take those two signals and align them in my brain, right? alignment in my brain. So half of each retina, right, is matching up with each other, and that's what that optic chiasm is, right? That optic chiasm is where the nasal or medial portion of each retina is crossing over to meet up with the lateral portion from the other retina so that we get one image from both eyes. Okay. So, again, as we're going from the eye into the brain, every sensory system, there's always exceptions to things I say here, but for your exam purposes, every sensory system is going to go through the thalamus, through a nucleus in the thalamus, on its way to its primary cortex somewhere in the brain. Okay? So the thalamus is going to be the first stop. Right? And for vision, that part of the thalamus is called the lateral geniculate nucleus, but we're going to call it LGN. And there are layers of the LGN that are receiving this input. It's the first place where the left eye, right eye images match up together in these layers of the thalamus. All right, so again, left visual field is hitting the medial portion of the left eye, the lateral portion of the right eye. Through the optic chiasm, those blue images are consolidated to the LGN, and then they go to the right LGN, and then they go to the right cortex. So my left visual world, even though it's coming into both eyes, is being consolidated contralaterally to my right visual cortex, right? And my right visual world is contralaterally being represented in my left visual cortex. So again, if you remember back to BioPsych, he talked about like the motor system and the touch system. Everything is pretty much contralateral in our brain, right? Switches over, so left side of the body, right brain, right side of the body, left brain. Here it's not so much side of the body as it's our world, a representation of the world, because it's hitting both sides of our body in terms of both eyes, right? Okay, so the first synapse in the central, so the ganglion cells that are receiving the output from horizontal, bipolar, and amacrine cells, those ganglion cells travel with their axons all the way to the LGN. That's the first synapse. That's one single axon from your eye all the way to your thalamus. Okay. Now, some of those axons peel off and go to the superior colliculus. What is the superior colliculus responsible for? Who remembers their sheet right here on anatomy? Here. Visual reflexes. In particular, eye tracking, eye reflexes. And so if somebody throws a ball across the room and you follow it with your eyes, you didn't necessarily say, hey, follow that, right? Your superior colliculus is a subconscious area that's doing reflexive movements of the eyes, particularly in response to tracking things or to honing in on things. There's also another part, and so we're not going to talk about the pretactile. I don't know why I have that on there. But I do want to talk about the hypothalamus because another small branch of optic nerve fibers go to the hypothalamus. Now, what's the hypothalamus involved with? What?
Memory.
Memory? Thinking hippocampus.
Is that the sensory relay station?
No, that's the thalamus that we're talking about. Hypothalamus, it is responsible for controlling hormones, so it's connected to the pituitary gland. It does a lot of things, right? Yes, Giselle.
Oh, I was going to say like the autonomic nervous system.
Yeah, so stress system is controlled by the hypothalamus. Hunger and feeding controlled by the hypothalamus, right? Why would we be sending nerve fibers from our eyes to the hypothalamus? What else do you think it's called? Sleep. Circadian rhythms, right? Sleep. So how do you reset your circadian rhythms? Light, right? When you have jet lag, if you remember talking about that in BioPsych, right? getting out in sunlight helps reset your internal biological clock. How does it do that? It's actually getting information about light directly into the hypothalamus. Okay. So these are all minor places. So just want you to be aware, particularly the superior colliculus and the hypothalamus, that some information from the eyes, from the vision system, peels off and goes to subconscious areas that we're not aware of in the brain. You're not aware of your circadian rhythm being reset. Subconscious. Okay. Everything before the optic chiasm we call the optic nerve. After the optic chiasm, we now call it an optic tract. That's sort of the difference in white matter that's in the central nervous system and white matter that's in the peripheral nervous system. Okay. Good? Cool, we're getting there. Maybe we'll see something today. All right, so we have these secondary pathways for, I guess I'm going to talk about the pre-tectum. Okay, hypothalamus, circadian rhythms, superior colliculus, automatic eye movements. The pre-tectum is part of the Edinger-Westphal nucleus, and the Edinger-Westphal nucleus controls our dilation and constriction of our pupils. What is that in response to? How bright light is, right? Dim light, we open up our pupils so we can absorb more light in. Bright light, we constrict them so we don't blind the receptors, right? So it needs to know how much light is coming in order to do that. So that's why that third thing branches off to the Edinger-Westphal nucleus. From the end, so it receives input about the intensity of light, and then it sends its output through cranial nerve 3, oculomotor nerve, back to constrict or dilate your pupil. That's the motor nerve that's doing that. Okay. Anyone want to guess what this little triangle thing is here? It's a space, actually. So if this is the superior colliculus up here, what part of the brain are we in?
Midbrain.
Yeah, the midbrain, right? And so what space or tube runs through the midbrain?
And dark ventricles.
The cerebral aqueduct, right? So there's CSF in here. And see the proximity, how close it is? That's why when you have drugs in your system, like alcohol, which is inhibitory, it inhibits the ability of the Edinger-Westphal nucleus to do this, which is why if you get pulled over for DUI, a police officer might shine a light in your eye and see how well your pupil constricts. If it doesn't constrict well, it's because you have enough alcohol here that's inhibiting the ability of this Edinger-Westphal nucleus to do that thing. So you're going to learn more than just neuroscience. Okay. All right. So we talked about all this. Get faster. The ganglion nerves cross over, right? And here's an important part. 1 through 5 in the left visual field, over here 5 through 9 in the right. That's a map, right? So this is reflected like that on the retina. We call that a retinotopic map, right? And that retinotopic map is conserved all the way back here to your visual cortex. So we keep everything in order from left to right in this pathway. All right, visual cortex. Yes?
So they don't ever, other than when they cross over at the chiasm, they don't ever become one picture until they get all the way back to back.
They start to combine in the LGN. I'll talk about that in just a second, a little bit more, in layers. And then, yes, those neurons then converge together to trigger one place in the V1. One place in V1. All right, so V1 is the posterior most part of your occipital lobe in the back of your brain. Right? Your primary visual cortex, we'll talk a lot more about that today. From there, it's also called striate cortex because there are actually like stripes from your left eye and your right eye. That's where those two signals are meeting up. So we call it striated cortex. After that, that signal is combined, and so you lose that stripe, And so we call everything else extrastriate cortex, which is your V2, V3, V4, V5, a bunch of parts of the temporal lobe, a bunch of parts of the parietal lobe. Visual processing is extremely complex. We use a lot of our brain. We use about 1/3 of our brain to see, right? If you said, what is the most important thing your brain does? You'd be like, think, personality, emotions, right? Those areas of the brain are very small compared to vision, right? Vision is so complex, we need a lot of brain power to do it. Okay. Thalamus. So, sometimes the thalamus is referred to as a sensory relay. It really is, a relay sort of says it's just handing the signal off. It is a filter. It is a sensory filter. It acts as a filter to let information through or to not let information through. And it does that for all of our sensory systems. It does the same thing for all of our sensory systems. It acts as a filter, not just a relay, right? And this is the first place that we can introduce top-down influences. What are top-down influences? So, what's that? Yeah, things, cognitive influences on our incoming bottom-up signaling, right? From prior experience, from arousal state, so particularly arousal state at the level of the thalamus, right? Have you ever been driving on a long trip and all of a sudden you're like, man, I don't really remember the last couple of miles, right? A little scary, but, right, sometimes you zone out and that visual information's not getting through, right? And so that can happen at the level of the thalamus, right? All right, so 1 million optic neurons, ganglion cells, right, come from the eyes, go into the thalamus, but actually 1.5 million leave the thalamus and go to visual cortex. So again, we are manipulating that bottom-up signaling. We're manipulating your visual sensations already before we even get to V1. So now we have more information leaving the thalamus than came in. So we're starting to diverge back out, right? So we talked about convergence as something that sometimes we have to do to get information into the brain, but once we get into the brain, we can begin to diverge out because we have many more cells in the brain. All right. So the brainstem involved with arousal also has connections into the LGN, and this is why your arousal state affects your ability to see things. Affects your ability to see things. Right? Study they did looking at this with sleep deprivation, right? So very simple study, there's a light that comes on. Every time the light comes on, you just press the button. Pretty easy, right? Light comes on, press the button. Light comes on, press the button. If you're like you are right now, where you're aroused and engaged and everybody's wide awake, hopefully, right? Then you're hitting that button every time. But if I sleep deprive you, all of a sudden you start not seeing when the light comes on and miss pressing the button. And that is because you actually, that signal's not making it up to your visual cortex. It's being sort of blocked or filtered at the level of the thalamus, right? Not something we really want to happen with vision, right? It's definitely not something you want to have to drive down the interstate, right? All right. The cortex can also supply positive and negative inputs for selective attention, right? And we'll talk more about selective attention in the next exam section when we talk about top-down influences, because we're focusing on bottom-up signaling, for example. Okay. So here's a picture of the lateral geniculate nucleus, and there's, as I mentioned, retinotopic organization of those incoming projections. So if we have ABC, they stay ABC, they stay ABC when they get over here. So there's A, B, and C stays in order like your eyes, like your retina, like your world, okay? you can see these C's and I's here. That's contralateral, ipsilateral inputs from each eye, right? So we're matching up the contralateral with the ipsilateral for A. So we get one representation of A, one representation of B, one of C. They're right next to each other. Right? There's 6 layers of the LGN, right? And so the ipsilateral is in layers 2, 3, and 5. The contralateral is 1, 4, and 6. Again, I didn't design this stuff. But 1 and 2 are fundamentally different from 3, 4, 5, and 6, which is why it's sort of different there, right? So the fact that like ipsilateral, contralateral is like this, and then it flips, and now it's ipsilateral, contralateral. It's because this area is called the parvocellular area, and this area is primarily receiving input from our cones. And where are our cones located in the retina? From the fovea. So this is our precise color vision. And again, you can see 2/3 of the LGN devoted to our center of vision. And then the magnocellular layer, the M cells, we'll talk about P cells, parvocellular, M cells, magnocellular, is coming primarily from our periphery, from our peripheral vision, and we devote less space to that. Cool? Okay. There's also some things in there called K cells. We don't really know what they do. Probably processing some of the visual signal. Again, what your brain is getting is very different than the actual light that struck your retina. Okay, so some things about P cells. Center of vision. They have a slow, steady response with small center surround receptor fields because we want good acuity, not a lot of convergence, right? Input from the fovea, from cones, color, and detail. M cells typically have more abrupt responses with large center surround receptor fields, and they're very responsive to movement. So sort of associate that M of magnocellular with movement. And so a lot of like what's going on in the periphery is we're looking for movement, and then the superior colliculus is reflexively putting whatever's moving on our fovea, shifting it so that now we're seeing what's moving, we can see what that is. So they receive output from the periphery, from cones and rods. They respond to movement and send signals to our superior colliculus and say, hey, put whatever that thing, I see something over there, look at it, identify it, put it in your fovea. Shift it from the M to the P. All right, so these are 2 sort of examples from your textbook. Sort of describing this. We have M cells here, magnocellular, parvocellular, movement, periphery, color, detail, right? And then these are projecting into primary cortex, and then from primary cortex we're going on to the secondary cortex. I don't want to spend too much time on that. This is a slide that I like. I think it's a little simpler than all of this stuff, which is that we have some neurons peeling off and going to the superior colliculus, but then we have 2 pathways out of the retina. We have the parvocellular and the magnocellular going into the LGN, right? The superior colliculus primarily getting information out of the magnocellular for that movement to orientate our eyes to things that are moving. LGN is the first place we combine ipsilateral and contralateral signals into one image. Okay? We have separation now of fovea versus peripheral with parvocellular and magnocellular. So we have detail and sort of movement. We have filtering that's happening in the LGN. And then we're going to hand it off to V1. And V1 is our primary visual cortex. Okay. So V1's primary function is to detect very primitive features in our environment. So you can think about our visual world's very rich and complex, and essentially what we're doing is breaking this complex picture down into very specific components that our brain is going to reassemble as our overall perception of vision. There are 250 million neurons just in V1, in each V1. So how many neurons are projecting into that out of the LGN? Remember what I said? Yes, what? This one? 1.5. So now we're going from 1.5 to 250. Big divergence. Big divergence back out into the brain. Okay, I'm projecting from 1.5 to 250. All right. After we get all these like details, angles of lines, colors, we have to reassemble them into a picture. We have to take all these little parts and put them back together as our perception, and that happens in the extrastriate cortex. So V2 and V3 are forming simple shapes from those individual components of vision, from those features. V4, we have a special area of our brain that's doing color perception, right? Laying color on top of this. V5, particular place in our brain for movement perception. Those are all the V's, V5. Okay. Now we have some other things like middle superior temporal cortex, MST, things in the parietal lobe, right? The temporal lobe is sort of complex object formation, right? And we'll look at how that gets broken down. But basically you have two output pathways out of the visual system. One that is ventral, that dives down into the temporal lobe, and what's located in the temporal lobe? The ethalamus, the hypothalamus. The hippocampus, and what is the hippocampus involved with? Memory, and where do we store memories? In neural networks in the temporal lobe, right? And so this is where identification and recognition is coming in, right? What is this? It's a cup. You recognize it as it being a cylinder, right? That it fits in my hand, it's a certain size, it's got a lid, right? And so you take all those features, assemble them back into the cup, and then send that information in the temporal lobe, and it goes, hey, I know what cups are. I even know what they do, right? Then we have a dorsal pathway that's going up into the parietal lobe, headed towards our frontal lobe. And what's our frontal lobe do? What's that, Ben? Executive functions, making decisions about stuff. And what else does a big chunk of our, sort of the back part of our frontal lobe do, right in front of the central sulcus? I heard someone say it here. What? Movement, right? So this pathway is, what do I do with this visual stimulus? This pathway is, what is that thing? This pathway is, what do I do with it? Do I need to make a decision about it? Do I need to make a motor response to it? What am I going to do with that thing? Okay? All right. Y'all know it's not something like that. Okay? So we have these center surround receptive fields. So here's two ganglion cells hooked up to our processing cells, right? So this ganglion cell has on center, off surround. This one has on center, off surround. They overlap some, right? They overlap some here, right? And so if we have light that comes in, it might activate this one, right? We have light that comes in here, it may activate these, right? And so because these things overlap, right, we're able to get more information. So here, for this one, all I know coming out of this is that it's inhibitory, right? But because this neighboring one is also activated is inhibitory, I know that the light is over here and not over here, even though it's only one inhibitory signal. So we can start to look at which receptive fields are activated in order to understand where that light is with the convergence. All right, so you can imagine our retina here, and we have a whole bunch of these donuts centered around receptive fields. And if I put more and more and more of them on there, right, we're overlapping these things everywhere, right? Complicated. What we start to get are strips or patches of excitatory surrounded by inhibitory, right? And we start to get those, we can get those at different angles. So we can start to detect orientation of lines by looking at the overlap of these, okay? So, in the 1950s, Hugh and Weasel, why are those names in red? Because you need to memorize them, right? Remember that MFT that you're going to have to take in your senior thesis? I bet these names are probably on there. You need to recognize these names and go, yeah, that was some of it with vision. One of these 4 multiple choice answers is about vision, right? We talked about looking at measuring the ganglion surround receptive fields in the retina of cats. They then moved back to the LGN and did the same sort of thing. They were putting dots of light on the screen, trying to map out the LGN to see the retinotopic map in the LGN, right? They won the Nobel Prize in Physiology and Medicine in the 80s for their work on this. But what happened was, while in the LGN and in the retina, they could map out these center surround receptive fields with these dots of light, right? When they got to the cortex and they started trying to do the same thing in V1, dots of light, weren't causing signals. They weren't able to find the receptive fields. So like, where did they go? We know the neurons go from the LGN to here, but we're not getting the same pattern of signals that we're expecting. And this is in the 50s. We didn't have fancy projectors like this, right? We had actual slide projectors that they were dropping a slide down in that had a point of light that was projecting onto a projector. And what they noticed was when they dropped the slide in that had a point of light on it, sometimes they would get a response in V1 as the slide went in, but then it would go away when the dot was there. And I don't know if it was Hugh or Weasel or both of them had the right idea that, hey, maybe we need to be not looking for dots of light, but for lines of light. And it turns out that when they switched to lines at different angles, they were able to get responses in V1. So V1 is coding for features, not just dots of light, but features like lines at different angles. And so they would find a neuron that had a vertical center surround receptive field, and when they put a vertical line in that receptive field, it activated all of these receptive fields, and they got a whole bunch of action potentials in V1. And as they rotated that line, now it's activating some inhibitory with the excitatory, not as much excitatory, and the action potentials went down. And if they rotated it perpendicular, they turned it off. Pretty cool, right? All right, so these are called simple cells. So simple cells respond to a specific orientation of a line at a particular angle at a specific place on your retina. Again, we have a lot more processing power now, 250 million neurons we can use. And so what happens is they could go in and measure, they'd stick the electrode down, they'd find one, and then this part of V1 was coding for a vertical line and not for another line, right? And as they changed that angle of the line, the more they got away from vertical, the less responsive that neuron became. Until at some point, it's rotated so much that it doesn't activate that neuron anymore. All right. Lots of examples of this. This is another example. This neuron is, this particular cell that we're recording from is tuned for a vertical line. That is its stimulus. Okay? The further away we get, the less action potentials, impulses go down. Okay. All right. So they stuck the electrode in, V1, and found that this cell was responsive here to a horizontal line. And the deeper they went in the cortex, they found that there was a column of cells all responding to horizontal lines. So they call these orientation columns, and it was for a horizontal line at a particular place in space that's projecting on our retina. Okay? Then, so here's a column of cortex that is coding, hey, there's a horizontal line right there in this part of my visual field. And if I move that horizontal line over here, I'm not going to be triggering this column anymore. It has to be at that particular place in space. Turns out if they took and went in, so they're sort of showing you if they go across the columns, the columns are orientated to angles of light changing in small increments. And so our V1 has a strip where for a particular place in space, every possible orientation of a line has a column that will fire in response to that stimulus. Yeah. So for a particular place in space that's projecting to a particular spot on a retinotopic map, okay, we have a group of columns that are coding for any possible orientation of a line at that place on our retina. So if there's a vertical line at that place on our retina, this column is firing. If we now change it to a horizontal line, this column starts firing. And that place in space on our retina is hitting two retinas. So again, striate cortex. We have a stripe for the left eye and a stripe for the right eye. So we call these ocular dominance columns. So an ocular dominance column is a set of columns that are responding to any possible line orientation in a particular place on our retina. It's a group of columns grouped together, adjacent to each other, side by side. And this group of columns over here is for the next little receptive field over in our retina. So that this is going from like left to right across our visual field in order of the retina, across these ocular dominance columns. This is how we break down this really complex visual stimulus into these features. Okay. So, if this spot here in green, right, is projecting into here, and just over a blue dot next to it in my receptive field is hitting the retina right beside the green dot, right? All of the information from the area of the retina that's green is coming in, left eye, right eye, ocular dominance columns, to tell me within this circle, a line that's vertical there, right, is going to cause this column and this column, right, because that's what's represented there, to be highly active. Right, to be highly active. And if there's an angle line there, that's going to cause maybe this one over here, which is for that angle of line. It's not here, it's back there, right? but it's a different place on the retina, but they're side by side. Do y'all get that? Do y'all understand that? Cool. I love this class. I'm a real geek about this stuff. I'm sorry. But think about how complex this is. And you're driving a car 70 miles an hour down the interstate, and your brain is like doing all this stuff simultaneously. There's no lag. Like, think about how much more powerful your brain is than a computer. A computer couldn't do that kind of processing and stay on the road. Right? It's amazing how our visual system works. All right, so we have these hypercolumns. A hypercolumn, it's like I said with the ocular dominance. So the ocular dominance means there's a left eye, right eye strip. A hypercolumn are those two ocular dominance columns combined into every possible orientation of a line at a particular place on both retinas. It's the same place in space. And we call this striate cortex because if you were to label the inputs coming from the left eye only, they would show up in these stripes, and in between these stripes are the dominance columns for the other eye. That's why they called it striate cortex back in the day, because it looked like stripes. Okay, so we have all these receptive fields. We've overlaid them all. These are donuts, center surround receptive fields, at the retina and at the LGN. But when we get to the cortex, we have so many more neurons, we can diverge and go from donuts into angles of lines. Right? So this is 25 ganglion neurons, all overlapping. Remember, we have 1 million of them. There's a lot of overlap, right? And they can project to create these. So, if we say that these are outputs, so I'm just going to make it a little bit more simple and just talk about the excitatory centers, not the inhibitory surrounds, right? But if these are all the centers, effectively, when I shine a piece of light on there, right, all of these 5 receptive fields come in and stimulate, all of them send an axon, they diverge from one into multiple different places, right? So that this column gets the most excitation when these 5 are all stimulated at the same time. That's how it codes for a vertical line. And if I rotate that line, right, now we took out the red neurons, right? But we added in these neurons, and so now they're hooked up to a different column, and the slightly angled line column becomes most active, not the vertical, right? And if we rotate that even more, right, we're sliding over in our ocular dominance columns, right? But this one, the yellow one, is part of a line that's centered on the green, right? So we're showing a line that we're rotating centered on the green receptive field, right? Which is why green is part of every column here. Because no matter what angle of line is stimulated, green's going to be activated, right? But yellow, while it's part of this angle, is also part of another vertical column down here, right? That when we put this, so their axons are going to multiple different columns, which then allows us to have a vertical line here that's now activated in a different adjacent part of the cortex that's saying, hey, this is for angles of lines centered here. This next column beside me is for angles of lines centered right next to me, and so on, all the way across the retina top of that.
So is this for each kind of like individual image, or is this more movement?
No, we're talking about details here, right? We're talking about breaking apart the complex image into its components, okay? So if you were to look at a picture and say, I'm going to draw that, you'd start with a line and add a line. You're adding lines, and that's essentially what we're doing in our brain. We are breaking this complex thing down into these individual lines, and when you group them all back together, you get the overall picture. Okay? So the overall picture, spoiler alert, is going to be looking at which columns are firing the most and grouping them all together with synchronous firing so that we can see the picture from all these individual lines. Okay, doing good? Halfway, kind of halfway. All right, let's see. Okay, so we talked about simple neurons. Simple neurons respond to a very specific stimulus and orientation of a line at a particular location on our retina, which is a particular place in space. Okay? And we talked about them as tuning curves because they're tuned to a particular orientation of line, and the further you get away from that angle, the less they respond. Okay? We talked about all this. We talked about this. Hypercolumns are the ocular dominance from the left and the right eye for that particular place with every possible line orientation. Okay. It's more complex than that, right? We haven't even talked about color yet, right? And color, I don't think we really understand how our brain does color. But, I mean, I'm sure there's probably some really smart visual neuroscientist that has a good handle on it. But colors is represented sort of in a weird way. It's not like, oh, here's yellow at this particular hypercolumn and red at this hypercolumn. It's representing what they call blobs because it looks like blobs. It doesn't really have a logical, for our brain to detect things. So going across this hypercolumn, right, you might have some of the angles and lines that are most responsive to yellow, but other angles most responsive to orange or red. Like, we don't want to think about hypercolumns like this. That's too much. We're going to think about them. We're going to take color out. And we're just going to be like, okay, we're going to talk about color. We're going to talk about V4 over here. Okay? But I want you to know color is represented as features in V1, but in a very sort of complex way across these hypercolumns. Okay. We have some other cells in V1 that are complex cells as opposed to simple cells. And complex cells respond to an angle of light, okay? But it's not at a specific location. It's not centered around a receptive field. It's that angle across a bunch of areas. So complex cells are orientation selective, like simple cells, right? But their receptive fields are a lot bigger. They're not centered around a particular place in our retina. So a simple cell would only respond to an angle of light here, and not if we move it over. But a complex cell would respond to that same angle of light even if it's moved over on the retina. But if we change the angle, it doesn't respond. Cool? Okay. Complex cells can also, not just form across a bunch of areas, but they also become motion detectors. They become motion specific. So now we have an angle of line, this angle of line, right, is most active. If I change the angle, cells stop firing, right? But it's this angle only if it moves in an upward motion. If I move that angle in a downward motion, it's not active. So now we've coded for an angle of light that's moving in a particular direction. So here we see this one's coding for a horizontal moving down, If I take a horizontal and move it up, no response. No response if I change the angle. So now we've gone from a simple cell into a complex cell that can code for an angle of light moving in a particular direction. Moving in a specific direction. Okay. Third group of cells are called end-stop cells. So these cells are going to measure for orientation of angle, but the length of the line. The length of the line now becomes important. How big is the line? So again, we have these tuning curves that it has to be this angle of line, and if it's this long, it's most responsive. If it's shorter, it's not activating all the receptive fields, so it's less responsive. And if it's longer, it's activating the inhibitory parts, right, and it's getting less responsive. So now, end-stop cells are cells that are tuned for certain lengths of lines to tell how long are those angles of light. Remember, 250 million neurons, we have a lot of divergence. We can send those simple donut receptive fields to lots of places in our cortex. All right. So, hypercomplex cells are end-stop cells that respond to movement. I'm going to stop there. That's like the most complex, because it's hypercomplex, right? Something more than that. Super hypercomplex. Yeah. Hypercomplex, okay? So now we have orientation of line, length, and movement. So if that length moves up, we don't get it. All right. Write down, slide 41, Dr. Pittman said this is a good review of these different types of cells. This is a good table from a different textbook that's not my textbook. All right, this would be a good thing to know, right? How are the receptive fields at the cell of the ganglion cell, the LGN, simple, complex, and end-stop cells? Okay. So now, here's what we've done so far, right? We've taken light to the retina, we've processed it to enhance edges, and we've converged it to go from a lot of receptors into a smaller number of neurons. We've gotten it to the LGN and combined the signal from two eyes into one spot. And then we've taken it to V1 and diverged it back out to lots of columns that are coding for lots of different features. And it's going to be the pattern of those columns firing together that we're going to reassemble in extrastriate cortex as your visual perception of objects and shapes and colors. We're going to reassemble that. Okay. So let's start with a very simple shape like this, right? Not really like things we look at in our world, but we'll start simple and go from there, right? So here we have vertical lines. They're going to be activating columns that are representing vertical. We have a horizontal line over here. That's going to be activating other columns. So when you look at this image, there's a pattern of columns firing that represents the shapes and lines and angles that are out here in our visual world. Right? So we can think about this in terms of, okay, let's look. Notice I took color out of the picture. We're trying to keep this simple, right? But we have vertical lines, horizontal lines, right? Curves. We have light areas and dark areas. And so intensity gets brought into this, right? And so there are other cells in V1 that help detect changes in patterns of light, because that can help us identify, hey, here's some changes in patterns of light that represent floors on a skyscraper, and they're not here, so I know the skyscraper stops there and doesn't continue over here. And so these are called spatial frequency detectors. And spatial frequency simply means changes in patterns of light over space. Frequencies over space. Here we have the same pattern of change, here and here. But here we have what's called high contrast, meaning there are very sharp edges in between those changes. And here we have low contrast, meaning there's blending between those changes. But the same frequency of change, right? So we can measure frequencies, how rapidly is light changing, and we can measure high contrast and low contrast. Right? So here we see low spatial frequencies, low spatial frequency, low spatial frequency, low spatial frequency. High spatial frequencies, changes in light, bricks, tiles. And then on top of that, we can look at high contrast, high frequency, right? Low contrast, high frequency, still high changes in light, but low contrast. Okay, so this sort of shows the whole spectrum. High contrast at the bottom. As we go up, the contrast goes away, right? The right is high frequency, the left is low frequency. And at some point, what happens up here? It blends together. We have a threshold for being able to detect this. And so actually, like you can see, like let's look right here. Can you see that there's change here? That's the same contrast as over here, but when we get to low spatial frequency, we can't detect those changes. We need the high frequency to see those changes in that contrast. So this threshold is actually like a curve, kind of like our color we talked about, where depending on the spatial frequency, depends on how easy it is to detect the contrast. Cool? Good? Okay, all right. So, I want to talk for a minute about adaptation. We're going to talk more about adaptation in lab. Where are you going for lab today? Down the hall. Don't come here. There'll be another lab. C25 down the hall. Okay. Lab today is going to be pretty short. I'm going to give you the instructions on how to do the dark adaptation lab, and then I'm going to turn you loose. It's going to take you about, you're going to work in pairs. It's going to take you about an hour for each person, so 2 hours per pair. So we're not going to spend 3 hours in lab today. It's going to be pretty short. You got to go collect your data before next week. Okay. On the website, there's a link to click to sign up for times to do this, so you may want to go ahead and do that. Okay. Adaptation. Reduced sensitivity due to repeated stimulation. We mentioned this last week. Right? The time course of this can range from milliseconds to much, much longer, depending on the sensory system, depending on the stimulus, right? So why do we have this? Well, one thing that can cause adaptation is our receptors get fatigued. If we're just stimulating our cones over and over and over again, we can deplete them of their photopigment. And if we deplete them of their photopigment, they become less sensitive. In fact, that's what happens in light adaptation versus dark adaptation. We have depleted all the photopigment from rods in bright light, right? And we have to build that back up in order to gain sensitivity. Okay? Our cortex, though, can also, if the same columns are being stimulated over and over and over again, they can sort of tire out and the signal starts to get smaller. And if that signal gets smaller, it's harder to perceive. It's harder to perceive in our brain. So we can have a sort of normalization response to the level of the cortex. And then finally, we can have the thalamus inserting its role, where it's sort of filtering things out based on attention. Based on attention. Or how prominent the stimulus is. Okay. And so, because we know how this stuff works, right, we can actually use adaptation to show that we have particular cortical areas that are responding to specific line orientations. Okay? So I want you to look up here. I want you to look right at this black bar. Okay? Everybody stare at the black bar. You're not moving your eyes. You're not moving your head. You're staring at the black bar. And then in a second, I'm going to change it. I want you to tell me what you see. So we're staring at the black bar. All right, what do you see? What happened to the lines when it went to a circle?
Straight.
So here, the angles of lines are this way and this way. And as you're looking at this thing, these parts of the retina are being overstimulated. They're getting fatigued. And then when I switch to vertical lines, you actually, for a second, get the opposite orientation and the lines kind of straighten back up. And that's because we have those tuning curves. And so if I'm stimulating you with an angle of line, and this is the tuning curve that's stimulating, right? We start to get adaptation. And so as I make you stare longer and longer at that center line, we're getting fatigue of those receptors, and this goes down, right? And these neurons are part of this vertical line column, right? So instead of firing up here, they're now firing down here. Well, when I now switch to a vertical line, because of this fatigue, these neurons aren't fatigued over here, right? So they start to fire more, and so you see the opposite angle over here until they recover. And as they recover, then that tuning curve comes back up, and you're like, no, that is a vertical line. It's not the opposite line. Did y'all get that? It's pretty cool. We can do like an illusion and prove to ourselves that we are reducing firing in columns in our V1 by switching to a different orientation and seeing that adaptation recover. So cool. All right, so look right here in the center of these lines. Just keep looking there. As you're looking there, are you seeing anything change? I'm seeing the intensity get like a little brighter and dimmer. It's starting to fade away a little bit. Back now it's starting to get really dim. Is that working for you guys? If I make it the same spatial frequency but lower, if you look at this one, nothing's moving on that screen. But as you adapt to that, right, the signal gets so low, if it goes below your threshold, you lose the ability to see those lines because of the fatigue that you're causing. Isn't that cool? All right. And it's orientation specific. So here, if we adapt to this and look at these lines, so stare, you got to keep your eyes still because we want to keep those spatial frequencies on the same places of our retina. But now you should start seeing it start to adapt some. And when I switch to this, what's your perception? Did you see hash marks until it recovered? Right? But there's really just horizontal lines there, right? Things changing there. That's sensory adaptation. All right. So if we have a picture of a face here, I don't know, why can they have them be like smiling or something like that? I don't know what the expression is. Again, I didn't write the textbook. But when we have a complex thing like a face, right, our brain is breaking that down, not only into horizontal lines, vertical lines, right, but also spatial frequency, high frequency areas, low spatial frequency areas, and laying that on top of each other, right? And this is how we take all of these features, all these individual features, and when we recombine them, we get this rich picture that represents our world. Cool? Okay. So we have some cells in V1 that are responding to different spatial frequencies. Again, this is all part of breaking down the image into its individual specific components and then reassembling them. High spatial frequency, low spatial frequency, all comes together. All right. Summary. Neurons in V1. Oh, so this is recording. Okay. Start with that.
More and more, we will be able to measure and understand how our brain cells function. Deciphering our consciousness is another question. But the experiments of Dr. Yang Dan have proved that in principle, it is possible to read and to understand the brain.
I didn't know that we started this data yet. So this is a digitized movie. We present this to the animal, and we record the activity of their visual neurons. And these electrical signals that we recorded from will travel through these cables.
She managed to tap the vision from the brain of the cat and to reconstruct what the cat sees onto a computer screen.
So this is a digitized movie. This particular movie is a short clip from Indiana Jones, I think.
Here we see the two images next to each other. On the left, the image that was shown to the cat. At the right, the image that is tapped out of the cat's brain.
The picture has a lot of random flickering, the noise.
Yeah.
That's probably the noise of the actual neurons. Because they sometimes fire spontaneous spikes. We consider those noise, but maybe they reflect something else, maybe the thoughts or something. But we can't distinguish that. So when we use all of those, thinking that they represent visual information, and this is the reconstruction that we come up with.
It is the first time an image was tapped from a brain. It proves that we can read and understand the grammar of it.
Oops, back up. Okay.
They represent visual.
All right. So they have hundreds of electrodes that are recording, but there are millions of columns of cells, right? So we're not able to get the full picture that our brain sees. So they're only getting small bits and pieces of the stuff that's coming in, but what they're getting are bits and pieces, features. Those electrodes are measuring individual columns responding to these changes in light, dark, spatial frequency areas. All right. So that's just an example. We actually we have science to back this up, right? What is this a picture of? Everybody get that pretty spot on, pretty quick? I mean, it's pretty messed up. Doesn't really look like him, but we are so good, right? Our brain is so good at reassembling that it's sending this pattern into your temporal cortex and saying, hey, does this match a face? And you're like, yeah, that's pretty. Now, if I were to go get a prospective student that's visiting campus and digitize their face and put it up here, you'd be like, oh, right? But this pretty famous face with pretty famous beard and everything, right? So you have a good memory of that, so your temporal lobe's able to recognize it. Okay. So a summary of this, we have simple cells, orientation, single feature. We also have spatial frequency detectors, complex cells, movement and orientation, end-stop cells, length of the neuron, movement and orientation, hypercomplex, right? Hypercolumns are a collection of all possible orientation columns in a particular place in space that are getting input from both the left and the right eye in that particular place in space. We good? Okay. Give me V1 feature detection. So we're just at V1. We're going to get out of V1 Thursday. Okay, let's see if we get the rest of this down. All right. So, if we look, so here's a cat that was raised only with vertical lines, here's a cat that was raised only seeing horizontal lines. If we look at what lines they can detect perceptually, behaviorally, or if we go try to map the hypercolumns in their visual cortex, they have only perceptions that are close to the thing that they experienced, and the complete opposite of that thing is not coded at all in their brain. Right? It's not coded at all. Now, where this where we have seen some of this is that there was a study that was done that noticed some vision abnormalities in infants that were in an orphanage that were laid in cribs, and the cribs had like vertical slats on them, and they just never got picked up. I mean, they just lay there all day looking at these vertical slats. And that caused in development difficulties. I mean, they didn't never see horizontal lines, but like deprivation, if you deprive someone of something, then it can affect the development of these sensory systems. But it really takes pretty severe deprivation for that to happen. All right. And so in that case with those infants, what happened was an early astigmatism happened. Sometimes people have astigmatism, but that not due to development, but due to misshapen parts of the cornea that actually cause you not to see those angles of lines as well, right? So early astigmatism, often we want to correct that with glasses as toddlers, right? Early on, so that they're seeing all of the shapes. Right? So when, I should have put this in here, especially since I had 3 minutes I could have shared with you. But when human infants are born, they're using their smell mainly to identify people and hearing. And their vision starts off with very low spatial frequency and gets not very good detail, and gets better and better and better across 6 to 8 months. That's about the time course there for that to happen in humans. All right, boom. Stay on top of these review points. It's a lot of material. It's 10 slides less than last week, but it's still a lot of material. So please learn this as you go. If you wait until the weekend before the exam, you will be climbing a mountain to try to learn all this. Please learn as you go. That's what I ask you to do. Right? This is what's important to take away from this lecture. I will see you all at 2:30. Down the hall, we'll have a short meeting